Deshmukh sir, President All India Association of Microbiologists, and Dr. Archana ma'am, President Delhi Unit MSI. I also welcome Dr. Akankshi Munjal, Incoming Assistant Professor, Department of Cell Biology, Duke University, USA. Now, I would like to invite Dr. Professor A.M. Deshmukh sir to speak few words to our audience. Sir, please.
Yes, now already I have talked, but as a President of Microbiologists Society, it is my duty to wish all the best to your continuous activities. Because without taking any rest, Delhi unit is continuously involved in arranging, and a better part in that, you are encouraging your alumni. That is, I liked it too much. Because now what you have taught in the past year, now you are watching them. That is the good part. And you are giving this opportunity to the students to update themselves in the subject. And as Akankshi ma'am has told, that she is talking little bit different from microbiology, but in biology. But let me tell again, no other sciences are separate from microbiology. There is a bioeconomics, there is a bio-business, there is a bio-battery, there is a bio-cement, there is a bio-steel, and many, many things I can tell. So biology is now closely related with any other branches, and Dr. Akankshi will explain the topic, I'm sure, in a simple manner, so that our students will easily get the knowledge and will update themselves. Thank you all for arranging such beautiful lecture. Thank you. Thank you.
Thank you so much, sir, for your encouraging words. Now, I would like to invite Dr. Archana ma'am to speak few words to our audience. Ma'am, please.
Thank you so much, Dr. Aarti. I am very, very happy, and I will speak in very short so that we don't have to wait more for Akankshi's lecture, and which is a very enlightening one on tissue morphogenesis. At the very outset, I would like to wholeheartedly thank Professor Deshmukh sir, because he has been very unwell in the past few days. But where there's a will, there's a way. He's always ready to come and join to encourage all of us. And also, if we are getting so much encouraged, then how much encouraged the younger lot, the newer microbiologists would be, I can imagine. And also, it's a matter of happiness that there are no distance barriers. She's speaking from the US, and we are keen listeners from here. I'm very excited about this lecture. In the end, I would like to congratulate Department of Microbiology, Institute of Home Economics, who is doing great in terms of providing very exciting intellectual material to all the students. And actually, this offer is open to everybody nationwide. And so congratulations. You're doing very well and coming together to organize such good talks. All the best, Akankshi. Looking forward, and all the best for your bright future ahead. We are very proud of you. Thank you.
Thank you so much, ma'am. Now I would like to invite Dr. Ashima Vora, ma'am, to introduce our today's speaker to the audience.
Yeah, good evening, everyone. I would like to introduce Dr. Akankshi Munjal to you. Dr. Akankshi would be an incoming assistant professor at the Department of Cell Biology, Duke University from January 2022. Right now, she is pursuing her postdoc from the Department of Systems Biology, Harvard University, USA. Akankshi has a number of fellowship and grants to her credit. She has published 6 research papers, and she has a patent to her credit. Then she is also a recipient of honors such as ISFS, which is Intersection Science Fellows Symposium Associate. She has also been awarded the EMBO Travel Award, which is very good, and she has a mentoring and teaching experience as well. She has presented her research experience, and she has been an invited speaker at a number of national and international conferences. And she is also a member of a number of bodies, international bodies like American Society of Cell Biology and Society of Developmental Biology. So it's very heartening to welcome your own student. It's a pleasure to have you all back sharing your research experience and knowledge with your juniors and of course with all of us as well. So I can only recall Akankshi as a very charming, cheerful and a bubbly child or a young girl full of energy and enthusiasm and a very, very keen learner. So it's my great pleasure to have Akankshi here. Welcome, Akankshi. And I would like to invite you to deliver your talk. Thank you.
Thank you so much for the kind introduction to Sunita ma'am for the invitation. I'm very, very excited to be here. I've tried to make things simple in the presentation, but I still encourage people to ask me questions on the way. So if something is unclear, please feel free to stop me and unmute yourself and ask me a question. Or I don't know if there is a chat box here that can be used, and then maybe
yes,
Ashima ma'am can.
Yeah, there is a chat box. If it is all right with you, we can have questions at the end of the lecture. Yeah?
Sure. Okay. Yeah, we can do that too. Yeah. So I'll just start, and ma'am, if I'm going over time, again, you can ask me like 5 or 10 minutes before to wrap up.
That's not a problem, Akankshi.
Okay. Okay. So I'll just start then. So I'll share a little bit of what I've done in the last, let's say 10 years since my PhD on understanding tissue morphogenesis. My broad interest is in understanding how complex three-dimensional organs with very robust forms, such as some examples are shown here, how they form from very simple embryonic tissues. And since the form of an organ is tightly related to its function, understanding the design principles of organogenesis is important, not only for healthy development and physiology standpoint, but also to improve our understanding of developmental disorders and pathologies. So over the years, different research communities have addressed this question to build a framework which can be broken down into 3 important parts. So the first part of this framework involves identifying developmental signals. So these can be growth factors. These are essentially genes that are encoding important proteins that can bind to DNA elements. They're either transcription factors or morphogens that pattern tissue. So for example, here is a tissue in orange, and it's patterned to express a morphogen or a transcription factor in blue here. So this patterning is then translated into forces that are coming from within the cells. And these forces are often produced by filamentous actin and non-muscle myosin motor proteins. So that's the second important part of this framework, where first you get the signal and then you get this pattern of intracellular forces. And these intracellular forces would then cause shape changes. For example, intracellular force in the center of this orange tissue here will result in bending of the tissue, and intracellular force in a polarized manner will cause this square-shaped tissue, for example, to extend. So I'm abstracting the problem here, but there are several examples that we see in different model organisms where this actually happens. So I'll give you an example from fruit flies, which is a very well-known model organism to study tissue morphogenesis. So here, the transcription factor snail and twist is required for ventral patterning of the embryo. Ventral is the side this is our front is our ventral side. So when the embryo is at this blastocyst state, which is only a collection of cells, you can specify that this part of the embryo will become the front of the cell. And these transcription factors then cause a collection of forces on the apical surface of cells. So epithelial cells have apical and basal polarity, and this transcription factors gives instruction to the cell that you need to have forces on your top. And when you have all these cells that have forces on the top, it results in a phenomena called apical constriction, where the apical surface of the cell reduces in size, and it results in invagination of this entire tissue. This entire tissue will then go on to become the muscle layer of the cell. So I'll let this movie play again so you see. So these cells are reducing their apical surface, and this entire tissue is invaginating and will go on to become the future muscle layer of our body. Okay, so this framework that I just described, where you have instructions coming from genes that specify where the forces should lie, which ultimately drive morphogenesis, actually has a lot of drawbacks. And the first drawback that we've noticed is that these forces that I just told you, actin and myosin, they're not static in nature. They're extremely dynamic. So using quantitative imaging, we can look at them live, and you will see that they are showing a lot of different behaviors. For example, here they are pulsing and moving in one side of the embryo. So this is an entire worm embryo. During my PhD, I found that this sort of contractions of actin and myosin are not given in form of instruction. There is no gene that is telling them, hey, actin and myosin, you need to increase and decrease to be able to cause cell shape changes. In fact, I found that these changes are a result of a self-organized behavior, where actin and myosin, together with its regulators that activate it, are causing this emergent phenomena which result in pulse contractions of actin. And this then drives cell deformation. So here, as we heard in the introduction, this is sort of touching upon that science is now without boundaries. During my PhD, I collaborated with the physicists to model this behavior and understand how physics can help us explain these contractions of actin and myosin. A second example where we're not relying on instructions from genes, but in fact self-organized behavior is during patterning. So there's a classic model in tissue morphogenesis which essentially says that, let's say we have some cells that are making a morphogen, and this morphogen will diffuse in space. And as it diffuses in space, cells will respond to the different concentrations. So here we have low concentration of the morphogen, and here we have high concentration. And depending on that, we can form cell type A. Let's say one will become neuron, one will become muscle, and one will become skin cell. So this is called the French flag model, and this is used a lot in the field of developmental biology to describe how different morphogen concentration can cause different cell types. But there is a lot of research that is now done to say that, the model is not as simple as is shown here. Actually, the gradient of morphogen is super noisy over time, but it is a self-organized behavior of the cells that causes them to form these sharp patterns of becoming either a neuron or a skin cell or a muscle cell. And this sorting behavior that the cells go through is actually very similar to how immiscible fluids separate. So, for example, how if you make an emulsion of oil and vinegar and shake it, you'll see that the oil and vinegar separates over time. So this is, again, we're using physics to understand how cells are behaving. Another example that's come out and that's beginning to be appreciated is the role of water in giving forces. So earlier I said that, most of the forces are produced by this actin and myosin. So myosin is a motor protein that contracts actin filaments. But a lot of recent studies have shown that it's not just actin and myosin that's making forces, but also pressure created by water. So if you pack water in a confined space, it builds hydrostatic pressure, because in nature you just don't find water alone. Water also has ions inside it. So there is an osmotic gradient, which along with the flow of ions, there is flow of more water in the tissue. So for example, here you see this is an embryo of a mouse, where these walls are cells, and there are ions and water in the middle. And due to osmosis, more water flows in, which causes this entire embryo to expand. And so this hydrostatic pressure has been known to drive tissue morphogenesis again in several different contexts. And this is like more recent work in the last 5 to 10 years. Okay, and I will show you an example where the extracellular matrix can also provide forces in driving tissue morphogenesis. And another thing I want to highlight here is that I drew this framework to explain how tissue morphogenesis is happening. But in fact, it's not a simple hierarchical pathway like this, where you have a gene telling what the pattern should be, and that pattern is driving tissue morphogenesis. But actually, in reality, because of morphogenesis, cells are moving, which causes the pattern to change, which will then cause the gene expression to change. So we have to treat these components not as hierarchical blocks of information, but in fact a network that is feeding back into each other. So in the next 20 minutes or so, I will try to convince you that an organ that I study will help us look at all these new behaviors that haven't been previously studied in the field of tissue morphogenesis. And this organ is the semicircular canals of the inner ear. So semicircular canals, these, as I said, they're present in the inner ear, and they're required for sensing balance and acceleration. So for example, when we move like this, there is a lateral canal moving, and then if this movement and this movement. So the way we are sensing all these three different movements is through the function of semicircular canals. Their structure is quite conserved across different species. So the main goal in developmental biology is that because we cannot use humans to study all these behaviors, we use model organisms. And the only way we can use model organisms is because a lot of these developmental pathways and processes are conserved across species. So the most commonly used model organisms are fruit flies, as I showed you, and worms, which are invertebrates, means that they lack a spinal column. And for vertebrates, we use zebrafish, frog, chicken, and mouse. So by vertebrates means that they have a spinal column. So they're closer to humans than fruit flies or worms are. So the structure of semicircular canals is conserved across all these vertebrate organisms. So we humans and fish and frog and chicken and mouse have 3 canals in each ear, but there are other animals that have 1 or 2 canals in each ear. Okay, so I study their development and morphogenesis using zebrafish. So before I tell you how the development of semicircular canal happens, I want to tell you why zebrafish is a great model system. So biology is going towards being very quantitative. So now that when we publish a paper, it's very important to show all the results of your experiments in a very quantitative manner. And zebrafish allows us to do that. So these fish can lay hundreds of eggs that are externally fertilized. So for example, in mouse, you have to kill the mother to get the eggs out. In zebrafish, you don't have to do that. They lay eggs externally, and they're extremely transparent, so we can look at them with a microscope without a problem. So here is a movie made by my colleague of the development of zebrafish. So here the embryo starts off as a single cell. You see 2 cells already here because they've divided. And you'll see that within 24 hours, this single cell embryo will form a fish-looking structure. So first the cells divide, as you see here. Then they will spread around the yolk. The yolk is behind. And then they will undergo lots of morphogenetic movements, which involves elongation in the anterior-posterior axes. So like anterior is like our head is anterior and our legs are posterior. So I'll just let the movie play again. So from a single cell, the first job of the embryo is to form a lot of different cells. And once you have a lot of different cells, you want to give them some information of axes. So you need to know what is the head and the tail, the left and right, the front and back. So this is what the embryo is doing right now. And then I will pause it. And you will see here that this structure is somewhat what a fish would end up looking like. Okay, so now the technology is so advanced that we can look at individual cells and we can mark out in an automated manner their boundaries and their membranes. And what we can then do with this information is that we can look at the expression of proteins inside each of these cells, so like in the blue cell or in the white cell over time. So we have the technology to do that. Another experiment we can do is if we want to understand the mechanics of the process, we can apply forces. So for example, here you can use a laser to cut the tissue and look at its relaxation over time. And the relaxation actually gives us some information of what are the forces in the tissue. We can also put stuff inside this tissue like oil droplets and ask, what are the material properties? So is the tissue stiff like a wooden table, or is it elastic like a rubber band, or is it viscous like honey? So we can ask all these questions. This is a very new technology. This came out a few years ago, is that we can look at the expression of all the genes at single cell resolution in time. So for example, each of these cells, we can now ask, what are all the genes that are being expressed by this cell at any given time? So for example, let's say a cell has 10,000 genes, but not all those 10,000 genes are going to be expressed at a time. Let's say only 500 of those are going to be expressed over time. With this technology, we can look at exactly what those 500 genes are in a given cell over time during development. Okay, so now coming back to the inner ear. So the reason why we think that fish is the best model system to study inner ear is because first, fish have inner ear. So here is a 2-day-old fish. It's super transparent, and the structure here is the embryonic structure that will give rise to the adult inner ear. So now bear in mind that we humans not only have inner ear, which has the semicircular canals, but also middle and outer ear. Oh, sorry. So maybe someone is unmuted. That's fine. It happens all the time. So this is the outer ear. So it's very hard to study inner ear in a lot of model organisms because it's hidden behind the middle and outer ear. But in fish, the middle and outer ear is missing. They only have inner ears. So we can look at the entire development of inner ear live without having to extract out the inner ear, the middle and the outer ear. So this is a major advantage that zebrafish offers compared to mouse or chicken. Another thing, it's a fun fact that I want to tell you, that zebrafish actually were found in River Ganga by the first biologists who started using it as a model organism for developmental biology. Okay, coming back to the inner ear. So in the lab, we use a a confocal microscope. I will not describe to you what a confocal microscope is. If someone is interested, please ask me and I can explain better. But we use a confocal microscope which allows us to give very high spatial and temporal resolution to look at the morphogenesis of different tissues. And in this case, we look at the morphogenesis of the ear. So morphogenesis of the ear starts off as a thickening, as this tissue, which is called the otic placode. So all our sensory organs, the eyes, the nose, the ear, they start off as tissues that are very simple. They are called placodes. And these placodes are specified, if you remember the framework that I showed you, these placodes are specified by developmental signals. So now this placode, due to influx of iron and water, will form this football-shaped structure, which is called the aortic epithelium. So here you can see how this tissue is expanding, and this is happening due to, essentially due to water. So if you remember in the introduction, I told you that water can create forces to deform tissue. So this is a really good example of how water can create forces for tissue morphogenesis. Now this tissue will continue to grow and form this super complicated structure, which is the semicircular canals. So I will explain the topology of the structure so it's easier to understand. So essentially, to abstract the problem, I will tell you that basically the problem we're trying to solve here, or the embryo is trying to solve here, is that how from a simple sheet how do you make a tube? So the way we can address this question is by different ways. As a geneticist, I can ask, oh, is there a tube gene that specifies this sheet to become a tube? As an engineer, I can ask, oh, what are the minimum things I need to fold this sheet into a tube? As a big data scientist, where you may have heard people are doing proteomics, genomics, I can ask, what are all the genes and proteins expressed in this tissue to make a tube? As a microscopist, I can just look at this transition and maybe see that, oh, first this square sheet folds and then you stretch it out to make this long tube. As a physicist, I can ask, what are the forces required to make this tube? What are the material properties of this square sheet of cells? And finally, as a statistician, I can ask if this process of transitioning from a simple square to a tube were to happen 100 times or 1,000 times, would I always get the same shape of the tube, or is there variation between each simulation? So in the field now of biology, again, it's not even biology. In the field of sciences, we have this approach called systems approach, where we rely on using all of these techniques to ask how morphogenesis is happening or how a biological phenomena is happening. So as I don't know if you remember, Asha ma'am said, I'm from systems biology department. Essentially what systems biology means is that you're not using a technique, you're not being a microbiologist or a geneticist or a physicist to ask questions. You basically rely on different techniques to look at systems level behavior or emerging behavior to ask simple questions. In this case, how do you get a tube from a sheet of cells? Okay, so now let's look at the inner ear formation. So here I've tried to draw a cartoon of what the zebrafish looks like, and we are putting it under the microscope where the objective is coming from the top. What I'm doing in this image here, this is a movie and I will play it, is that I'm acquiring sections, optical sections, and I 3D reconstruct them. And if you remember, I said that the tissue that gives rise to the semicircular canals is this ball-shaped epithelium, and we're only going to look at the top half of this ball. This is where all of the morphogenesis is happening. Okay, so here is that outline of that ball. It has a lumen, which I said is full of water and ions, and it's sandwiched between the hindbrain, which is present here, and skin, which is the outer layer. So morphogenesis begins when 6 regions in this tissue begin to protrude or bud into the epithelium. So here you'll see that these buds form first, and later on there is a bud that comes from this side. So I'll let the movie play again. These buds extend towards each other. So for example, this bud in the anterior part will meet this bud in the lateral part, and this bud in the posterior part will meet this bud here, and they fuse to each other, as you see here. So this fusion results in these structures, which are pillars. So these are literally like pillars outside the house that define the foundation of our house. So they form these pillars, and the negative space around them corresponds to the semicircular canals. So the topology will become clear from these cartoons. So here is the aortic vesicle. So I showed you 6 buds before, but I'm only going to show 2 buds in this cartoon. So these buds form, they extend, fuse to form pillars, and the space around them, so you see this looks like more like a tube now, corresponds to the semicircular canal. So when we move, the fluid that runs in this tube also moves, and this mechanical signal is actually transduced by hair cells. I don't know if you notice. So these are the hair cells. These transduce the fluid mechanical signal into electrical signals, which are then passed to neurons, and this is how we sense motion. Okay, so now the question that I ask is, what is the mechanics of this process? How is morphogenesis happening of this process? So here I want to highlight that this is a multi-scale problem. We cannot ask this question by simply looking at either just the cells or the entire organ or looking at just the genes. We have to look at it from many different angles. And before I go into showing you some data, I want to highlight that I will go back and forth between images where you'll see either these 3D reconstructed tissues, or you'll see only one section, so one slab of this 3D reconstructed tissue where the buds are forming in different planes. Okay, so in the field, there are many different hypotheses of how you can make a tissue, a flat tissue into this bud shape. For example, you can say that, oh, maybe some cells are locally proliferating such that they bend to form this kind of bud. Or there's this phenomenon called convergence extension where cells exchange neighbor to result in this bud-shaped structure. In physics, there is a phenomenon, for example, if you have a straight rod and you're pushing the straight rod from both sides, it's called buckling, and this will cause bending of the rod into this tube-shaped or bud-shaped structure. And another hypothesis is that something is happening locally to these cells here that causes them to form this bud-shaped structure. What I found was that actually something is happening locally in these cells here that causes them to form a bud-shaped structure. And the way I did this was that if I kill, so I can use a laser ablation to locally kill a single cell in a bud. If I kill a single cell, it doesn't block morphogenesis of, it blocks morphogenesis of this bud, but it doesn't block morphogenesis of the adjacent buds. They continue to form and grow. So to dig deeper into how the morphogenesis is happening locally, the way we do it is we go and ask literature, is there something known in the literature? And in fact, almost 30 years ago, and this experiment was ignored for 30 years, there was a paper published in the journal Development, which showed that there is a component of the extracellular matrix that is required for morphogenesis. So what is this component? So before I tell you about this component, I want to tell you a little bit about what the extracellular matrix is. So as you may have read in a cell biology class, epithelial tissues usually sit on the basal side on a basement membrane. And this is composed of very stiff, long proteins that hold the integrity or hold the tissue in place. And we all have it. So underneath our skin cells, there is a layer of this basement membrane. But the component that is driving morphogenesis of the inner ear is not a proteinaceous proteinaceous component of the extracellular matrix, but in fact, it's a sugar. It's a polysaccharide. So this sugar is synthesized in the cytosol, and it's polymerized by these transporters that sit on the plasma membrane and extrude out large chains into the extracellular space. So we now have techniques to look at individual genes inside the cell in a very quantitative manner. So I looked at the genes that are synthesizing this non-protein ECM using fluorescent in situ hybridization. Again, I will not go into the technique here, but I want to expose you to the kind of technologies that we have. So using fluorescent in situ hybridization in a quantitative manner, we can look at, so this is the tissue, and only the cells that are budding, as you see here, in time, are the ones expressing the genes for ECM. So you see them in green and magenta here. And using either a morpholino, which basically is an antisense RNA that binds to the sense RNA, or CRISPR-Cas9, which you may have heard, where you have the Cas9 protein making indels or chopping off DNA at the site that you want, we can create mutants where essentially the function of these genes or the expression of these genes is blocked. So this gene was required for hyaluronan synthesis, and we now have a mutant or a knockdown using CRISPR of these genes, and we see that in controls, the development is happening normally, but in these knocked out embryos, the development is completely abrogated. Okay, so now I told you a little bit about the genetics. Now let's ask the physics of the process or the mechanism of this process. So how is this extracellular matrix component hyaluronan driving morphogenesis? So as I said, hyaluronan is a non-protein component. It's in fact a sugar. And this sugar has negative charges on it. So it's anionic in nature, which means that as it's made and secreted into the extracellular space, it attracts sodium ions and water to form a swollen hydrogel. So a good way of thinking a swollen hydrogel is agarose. So this is what you use to run a PCR gel, right, in the lab. So it's very similar to that. And this hydrogel has unique properties. So this means that depending on its the concentration and the molecular weight of hyaluronan, you can make a hydrogel that is not only stiff, so it's not only stiff like a table, but also viscous, which means it can flow like honey. So this is a very unique component that you're using now for morphogenesis. And to put it into context, this hyaluronan component you may have heard is used a lot in skin treatments. So skin creams have it, and the properties are also very similar to how the material used in diapers and sanitary napkins, where they can hold a lot of water in place. So here essentially, biology or naturally, we all are producing a lot of hyaluronan for joint lubrication and so on, but the embryo is also producing hyaluronan to drive morphogenesis. Okay, so using a binding protein that has high affinity for hyaluronan, I find that hyaluronan is enriched in the extracellular space underneath the cells that are budding. So this is similar to doing an antibody antigen staining, but here remember this is not a protein, so I'm not adding an antibody. I'm adding a binding protein that recognizes hyaluronan. I'll just skip this slide. So what I see during morphogenesis is that as these cells begin to bud, they're making large amounts of this hyaluronan, which causes swelling because of osmosis, and then drives morphogenesis of the overlying tissue. And I can digest this matrix using hyaluronidase, which breaks down the polymers into oligomers. And when I do that, you can see that this extracellular space actually now completely collapses, and the morphogenesis fails to happen. An important thing to bear in mind is that the kind of experiments I show you, we have a control condition and a perturbed condition. This is how we do experiments in biology in general. So earlier I showed you mutant and CRISPR mediated knockout, so where I showed you control and mutant, and here I'm showing you control of what is happening normally, and then I'm perturbing using this hyaluronidase. So this is how we check if component A is required in a certain process, is by removing component A or overexpressing component A. Okay. Another thing that I observed is that, again, I want to show you how microscopy is helping us a lot to observe all the processes, is that as these cells are making buds, they're stretching a lot. So they are really behaving like an elastic rubber band, where you see that they are stretching in this longitudinal and circumferential axes and thinning in this radial axis. So this is like a cross section. If I do a cross section here and I look at the cells from the top, you'll see that basically from fatter and like a small tissue, they're stretching out into a thinner tissue. So this is similar to, like, imagine you have clay in your hand, and when you, or a dough, and you flatten it out, so it thins in the radial axis, and you can stretch it out in longitudinal and circumferential axes. And this stretching that the cells are experiencing is in fact dependent on this ECM component hyaluronin, such that when I inject hyaluronidase, which breaks down the polymers into oligomers, you'll see that the stretching that the cells experienced is now lost. So here again, I'm doing a loss of function experiment where I'm asking, what happens to these cells if the component that is causing the stretching is missing? Okay, another thing we observed is that, which is very common in the field of morphogenesis, is that morphogenesis is often polarized or anisotropic, which means that when you create a deformation, it's not evenly spread across the tissue. Some cells experience more deformation compared to the other. So here, instead of making an isotropic bud, like a dome shape that you see on top of mosques, it's not a simple dome shape, but in fact, the bud is extending in a polarized manner into a tube. So we ask, where is this anisotropy coming from? Is it hyaluronin, which is this ECM component that is giving the anisotropy? So this was our first hypothesis, and we ruled out a hyaluronin-based hypothesis, primarily because hyaluronin is driving morphogenesis through pressure from water. And this is a classic law in physics, which essentially says that it's Pascal's law that pressure for a fluid is isotropic. And instead, we looked at these epithelial cells that are undergoing deformation and asked whether they are causing anisotropy. So epithelial cells can sense, respond, and produce forces through their cell cortex, which is composed of filamentous actin and non-muscle myosin motor proteins. So this is something I mentioned at the beginning of the talk. The field of morphogenesis for many, many years had been dominated with this idea that you can create forces with actin and myosin. And so for example, we have a cytoskeleton, we have a skeleton like the ribs and the bones that we have everywhere. The cells have their own cytoskeleton, which is essentially actin and myosin and microtubules. So I looked at where actin and myosin is present during morphogenesis. So normally actin and myosin is present on the apical surface. So this is the surface which has the fluid. But during morphogenesis, they are present on the basal side. So this is where the extracellular matrix is present. So these are static images, but we can also look at live using high resolution microscopy. And here you see that actin and myosin are not just present statically, but in these dynamic protrusions that are coming out of the cell. So here you see these protrusions forming and retracting. So this is something, again, I want to highlight here, that these static images that we see in textbook can sometimes mislead us into thinking that everything in biology is static. In fact, it's the opposite, that proteins are moving. Cells, as I showed you, are very dynamic. They don't have straight membranes the way we're used to thinking, but in fact, their membranes can have very unique features. Like in this case, they're making these tethers with protrusions from neighboring cells. And in fact, these tethers can have a lot of information in them. They can transmit signals to other cells. By making these tethers, they can transmit forces as well. So again, just we shouldn't think of cells as static, straight structures, but as these dynamic structures. So I found that these protrusions, they are naturally retracting as the ECM is spreading. So you can look at this animation where essentially now, let's say we have a balloon. In this case, the balloon is filled with this ECM component that I just mentioned, and it's expanding due to osmosis. And as this expands, anything if there is a rubber band around it, that rubber band will retract. I'll just play that animation again. That rubber band will retract, and this is exactly what's happening. You'll see this tether retracting. So this retraction is a signature that these rubber bands are in fact carrying forces. They are carrying tension. And what we find is that these tethers are in fact not distributed symmetrically, but rather asymmetrically, which means that they have a higher distribution in this circumferential axis. So now I told you, think of these cells as like a dough. And now you're trying to roll the dough. Let's say if you want to make this layered roti, I can't remember what it's called, but you can roll the dough into a very long structure. So you roll the dough. So your hand is doing this asymmetric movement, which is constraining the expansion of the dough in this axis and only allowing extension in the orthogonal axis. And this is similar to a corset too. So for example, if you wear a corset dress, you can cinch it in this direction, which causes expansion in the orthogonal or the opposite direction. So this is in fact what we see, is that these tethers that I just showed you are asymmetrically distributed in the circumferential axis. So again, to highlight that we can ask a lot of biology questions using physics and math. I collaborated with a physicist who made a simulation where he essentially added theoretically these inward forces that are causing constraint in this circumferential axis and expansion in the orthogonal axis. And in this case, if he removes this constraint, you'll see that the shape becomes very isotropic. It relaxes into an isotropic structure. So I'll let the simulation play again. So now this is only a model. This is again something important to keep in mind, that as a biologist, if I talk to a physicist and say like, hey, I observed this phenomenon, can you see using physics if there is any theory that can explain it? They can come up with models, but these are models. You have to test them experimentally. So I experimentally tested this model to ask that, what if I remove these forces now that are constraining expansion? And so here is so I use an inhibitor of F-actin polymerization. This is a very common experiment done in cell biology and developmental biology, where you can use pharmacological drugs, inhibitors against proteins. In this case, I'm using cytokinesin D, which inhibits F-actin polymerization. And when I do that, maybe you'll be able to appreciate that this anisotropic tube now becomes like a dome-shaped structure. So what I've told you so far is that certain cells in this tissue are patterned. Remember this framework where there are developmental signals. They pattern this tissue to synthesize a unique extracellular matrix. And this extracellular matrix then swells because of osmotic pressure to drive morphogenesis of the overlying tissue into a bud. And the tissue, in return, is making these actomyosin-rich tethers that constrain the pressure in this circumferential axis and allowing extension in the orthogonal axis. And this model that I show you here is very different from the classical model that the field of tissue morphogenesis has, where there essentially you can explain all morphogenesis, such as bending or elongation or tube formation, by just intracellular forces. What I've showed you instead is that actually the forces can come from outside, in this case extracellular matrix, but also from water. So for example, you can have a tissue with uniform force. So if you have a circle, you expand it into a bigger circle. If you have a hemisphere, you make it into a bigger hemisphere. But you can combine these isotropic forces with anisotropic stiffness. For example, if I constrain the expansion of circle in this axis, it will form an egg-shaped structure. See, this looks like how a chicken egg would look like. Or if I constrain the expansion of this hemisphere, it will make a bud or a long tube. So this is the new emerging framework of tissue morphogenesis that me and many of my colleagues are now working towards. The other thing I just want to touch upon in the last few minutes is that is the genetic component of this process. So if you're in the field of developmental biology and you start talking about physics and forces, people look at you and ask you like, hey, but what about the genes? Like, we only care about like, if this gene is removed, will it cause a genetic disorder? So for example, the gene that causes synthesis of ECM, if you remove that gene, it can result in congenital defect. For example, it can lead to inner ear disorder or imbalance disorder. So for example, a common semicircular canal defect can cause vertigo, which I'm sure you've heard. This is the spinning feeling when you are at higher altitudes. So to address what are the genes being expressed by these cells, we use this genomics approach, which is essentially I spoke a little bit about in the introduction, is that we can ask what are the genes being expressed by each of these cells at different time points. So what we do essentially here is that we take this tissue, which is the inner ear tissue, dissociate it into its individual cells. So essentially we add trypsin and a lot of enzymes. So essentially from a big block of integrated tissue, you have individual cells, and we pass these individual cells into this microfluidics device. And this microfluidics device is essentially barcoding each of these cells. So this barcode is very similar to the barcode you find on the products you buy at a grocery store, where you essentially can scan and say like, oh, this barcode corresponds to this being ketchup or Maggi. So essentially we're adding this barcode in the form of DNA elements. And now we can sequence this barcode using RNA sequencing and find all the genes being expressed by individual cells. So here each dot is a cell, and it's in this high dimensional space where it can have where you have information of all the genes being expressed by it. We can do a lot of analysis to reduce it to essential components that are, so let's say I have 100 cells and I want to categorize them into skin cells or neurons or muscles. So these are the three categories that I want to get. How will I do that? I know that skin cells should have protein A. I know that neurons should have protein B, and I know muscle cells should have protein C. So essentially, this is what we're doing here. Because I know from this technique which proteins or which genes are being expressed, I can categorize them into these individual types. So using this technology, we have information of all the genes being expressed by this tissue during morphogenesis. And now we can ask, okay, what happens if I knock out gene A? What happens if I knock out gene B? So not only will it tell us about how healthy development is happening, but it will also give us information about genetic defects and developmental disorders. Okay, and finally, in my own lab, what I will study are the unique functions of the extracellular matrix. So I just want to highlight here that the extracellular matrix is not well studied in the field of tissue morphogenesis, and this is something that I will carry out in my lab. I will also study how feedback interactions happen. So remember I told you that this hierarchical pathway that, oh, you have a gene telling this is the pattern, these are the forces, this is what you're supposed to do. It's not hierarchical like that, but there are lots of feedbacks that make the network robust. So I will study what are those feedback interactions, what happens if you remove one component, how are the other components affected. So these are the kinds of questions that I will study in my lab. And with that, I'd like to thank all these people who've helped me that are in my current lab to carry out my project. And I want to tell you that I will start my lab at Duke University in January, and I will be hiring at all levels. So if you want to apply for a PhD program at Duke University, or if you want to do an internship, you can write to me and we can discuss possibilities. And I'll end here and take questions.
Okay. Yeah, thank you so much, Akankshi, for that wonderful lecture. So I have a question, Akankshi. You said hyaluron is a major constraint, like it is essential for cellular morphogenesis, right? So
yeah.
What triggers its synthesis? How does a cell come to know that, yes, it has to synthesize this component so as to undergo budding and morphogenesis? So
that's a great question. I don't know the answer to that. And that's exactly why I did this RNA sequencing experiment, where essentially I know like all the genes being expressed by the cell at that time. So I'm looking for an important transcription factor now, for example, that can say that, okay, this transcription factor should result in expression of hyaluronan, but I don't know the identity of that transcription factor yet. But you're absolutely right in asking the question, is that how does the cell know that it needs to express this? I just don't know the identity of that. And that's why we did the final experiment that I showed. And I have a list of genes that are candidate genes that I will test now. So coming back to like, how do you test these things? You knock them out. So I will test them by doing CRISPR of each of these genes and asking whether hyaluronan is still being expressed or not.
Okay, thank you. But let me compliment, the videos were very nice and they explained the concept very well.
Thank you.
Thank you. Any other question anyone wants to ask? You may please ask.
Akankshi, it's really a wonderful talk which you have given. You have said, you're talking about this hyaluronan. Is beside this morphogenesis, does it has any other function also?
Yeah, very good. Yeah, it's a very good question. It does actually. Every time we have an injury, there is a swelling, right? And that swelling is actually partly caused by hyaluronan, because it's trying to create a cushion or a medium there where a lot of cytokines and it's a very dense matrix, so it can trap a lot of signaling molecules. So it's used. It's a response to inflammation. It's also there in our joints. So in our knee joint, when people have knee issues, they actually surgeons they inject hyaluronic acid to improve lubrication. So actually, its functions are very well known in adults, but not so much in development. So when I started studying its function during development, the literature was very little. Most of the literature comes from these orthopedic surgeons who use it in bioengineering and skin treatments for lubrication and inflammation. Yeah. And another actually well-known function is that cancer cells, when they undergo metastasis, also produce hyaluronan. Okay. So it gives them soft surface to crawl on. So that's another place where hyaluronan is very well studied.
So does there any way to increase its production during the aging process, because it may help in the joint problem?
Yeah, actually, I mean, you're asking if you can genetically increase it. I think the easier way is to use synthetic approach. So people, because you can synthesize, it's just a sugar, right? So it's not like a protein. You don't need to have bacteria expressing protein. Because it's a sugar, you can synthetically, like glucose, make tons of it. And so during aging, people actually, surgeons inject it rather than normally producing more of it. Because during aging, if you're asking the question if we can physiologically produce it on our own, you still have to regulate the right cells producing it at the right time. Because as I said, like even cancer cells can produce hyaluronin, so you don't want all the cells producing it. So it's better to use surgeries where you can locally add it where you want it.
You still are working on this zebrafish or other models you're using?
No, I will continue only with zebrafish. It's actually very intensive to keep a zebrafish facility. It's expensive. So like where I'm going in Duke University, they already have a zebrafish facility. Flies are easier to use. So if we have some experiments that cannot be used, that is difficult to do in zebrafish, you can instead use fruit flies also to address them. But remember, like fruit flies, they don't have inner ears like us. They actually use this haltear to balance. So there are a lot of things that you can't ask in other model organisms. But yeah, I will continue with zebrafish.
Okay, thank you.
Okay, so
it was a very good talk, and all the best for your future work. God bless you.
Thank you, ma'am. Thank you.
Akankshi, we have one more question from our second year student, Yukta. Yukta asks, does abnormal tissue morphogenesis leads to cancer?
Yeah, that's a very good question. Can cells? This is an important question in development. So one thing I didn't show you is that so epithelial cells don't necessarily stay epithelial throughout. They can change to become mesenchymal, which are more migratory in nature. So epithelial cells are like, if you want to imagine what they look like, they're really like our skin cells, where you see these hexagonal shapes, and this is how they maintain the integrity and form barriers. So they're there in our intestine, they're there in a lot of places. But what happens a lot of times when epithelial cells are dividing or undergoing morphogenesis is that if they receive wrong information, or if they have some misplacement of proteins, instead of being epithelial and part of the tissue, they can escape the epithelium and become migratory. So it's not so much that during tissue morphogenesis can cells become cancerous. I think cancer is a more aging-related process. Like during development, it's actually a good question for me to check too, that can cells become cancerous during development? But we can use this phenomenon where epithelial cells sometimes lose attachment to the tissue and become migratory as a model to understand cancer. Because this is what happens during cancer metastasis too, that these cells become migratory, they go to places that they're not supposed to be in and start dividing and making tumors there, right? So this is how cancer progression happens. So we can use what's happening normally during development to ask that question. So I'm just like changing a little bit what you ask and saying that how that same thought can be used to understand cancer.
Can they ask any question related to their career path?
Yeah, of course, absolutely. I'm happy to share anything regarding my experience or trajectory.
Girls, you can ask, beta. अगर कोई कुछ आपको पूछना है तो you can ask.
I also I think I can highlight here that you've there's a lot you can do with microbiology. When I started after my bachelor's, my master's project was on an HIV encoded protein called NEF, which actually allows HIV to escape immune responses. So it downregulates cell surface proteins like CD80, CD86, which interact with of making this immunological synapse. So it actually removes them from the cell surface, and then this is how like T cells don't know that, oh, there are some cells infected, so they can't basically remove these infected cells. So this is how I started. But then when I was doing this project, there were other people around me who were doing very fundamental cell biology problems in the lab. And this is how I got attracted to cell biology and developmental biology. And then I changed my career path. So that's something that is like very common in science. Like people change career paths a lot. They feel that they can be doing one thing during their bachelor or master or PhD and completely different during their postdoc. So if you like something now, that's good. If you don't like something now, that's also good, because there's a lot that science has to offer, and you don't have to stay in that boundary for sure.
True.
And actually a life-changing sort of experience was to go to Indian Institute of Science during my bachelor at IHG. So I did a summer internship at IISc for 2 months, and I applied to this Indian Academy of Sciences scholarship. And this gave me a really good exposure to research science, and I was in a developmental biology lab where they worked with dictyostelium to study dictyostelium life cycle. So that was a really good exposure for me to do research. So I think like I would really encourage people to look for these internships in labs. This is very common in the US, where people do more often in India during their masters, where they do 6 months internship during their masters. It's not so common during bachelor, but I would encourage people, if they can find, to do this during their bachelor too.
They can apply to your university during the summer.
It's harder to get people from India here because of visa and like funding, because you need a much higher funding to get someone from here. But there are a lot of great labs in India too, and I'm happy to share like places that host undergraduate bachelor students in India too, because it's easier for people to go. At the level of masters, it's more feasible. And PhD at the level of undergraduate, I think like even within India, like NCBS or ICERS, they have a lot of opportunities.
Akankshi, your talk was a great talk with very good life examples, like you said, flowing like honey. So just giving you one example, so you can immediately visualize that, and it gets inside your head also as an audience. So that's a great way of explaining something. And I would like to say to all the students present here, and even if they couldn't make it or they left in between for some urgent work or anything, we could just circulate your talk as a YouTube recording, and we would send you also through Dr. Sunita. I would like to say that such talks, besides imparting the knowledge, the current level of research, the global research, and the latest developments in these fields, besides that, it also plays a role that children can look at their role models, their seniors who have gone through the same path, and they have risen to such a level. And you're such a young girl, but explaining things so lucidly with all self-explanatory slides and videos. So, and your talk was a change of flavor for us because we don't have these things in our curriculum.
Yeah,
so
totally agree with Dr. Archana. Yes.
Yeah. So that is why it was a very enriching experience. You did it perfectly. I would give you 100 on 100 for that, because because if you can do this at such a young age, what are you in when you reach our age? So it's a treat for any teacher. I can really relate to it. And मुझे तो पता नहीं क्यों connect लग रहा है बहुत ज़्यादा.
She's going to be an excellent teacher. I'm sure she's going to be an excellent teacher. The way she's presenting her work, the way she's presented, and the way she speaks, you're going to be an excellent professor.
So she's going to join as incoming assistant professor. So now she knows where she stands, and this should boost her confidence as well. And in the end, I would like to say that the teachers who invest their labor, their interest and time and efforts and prayers and whatnot in their students, there cannot be anything more rewarding than this kind of outcome. So I really congratulate all your teachers. And so it is a very heartwarming thing. Thank you. Thank you, and thank you, your teachers.
Thank you to the teachers here. Yeah, not thank you.
The graduation also, she was always interested.
I wanted to say this, कि teachers तो अच्छे हैं ही, वो तो हम जानते हैं. हाँ, ये so like for me, they are my seniors, juniors and friends actually, close to heart. But ये भी एक साथ add करना पड़ेगा. We are the same for all. But it also depends upon the student who has faith or not, who takes us seriously and our effort seriously or not, which is written on the wall that you did, which is why you are doing what you're doing. So very, very happy for you. Very, very happy for you.
Thank you.
God bless you.
Thank you so much. Thank you. Yeah.
Thank you.
No, this was. I'm really grateful to Sunita ma'am for inviting me. We've been trying to coordinate since last year. I'm glad.
It's a pride for us.
Sunita, I had also planned a talk for today, but when I saw your notice, I postponed mine because I wanted to listen to her.
Earlier, it was planned for 19th, but because of some reason, for 18th.
So when I saw the revised one, and the next one is also on prebiotics and probiotics, so I am steering clear of all these slots so that I can attend all. It was worth it. It was worth it. And I would recommend people to go through it again, actually. And good that we have got the recording. And I owe my gratitude to my student Vishal, who is always available. And even if he has some other work, he somehow accommodates. So thanks to him, it is available to all. And Akankshi can also get it, and she can also look at herself. And it was very effective.
Scary to look at yourself talking.
No. It was very impactful and very effective. And it was a treat, I would say.
Thank you. Yeah.